I'm here with Sai Garte. Hello, Sai.
Hello.
Nice to have you at my channel first time, and it's a pleasure to have you here and to talk a little bit about abiogenesis, evolution, origins. I know that you are a Christian and evolutionary biologist, and you're a theistic evolutionist. Is that correct?
Yeah, more or less.
Okay. So before I went live, you started to telling me about your recently published paper. Maybe you can go on and tell a little bit about it.
Sure. So what happened, you asked me what my research fields are, and I mentioned my research fields when I was an active professor doing research. But then once I retired, I began pursuing an interest I have in, and I've had for a while, in the whole issue of origin of life. And a lot of people, of course, are doing research in that field, but my interest is very specific, and it has to do with the question of self-replication and the accuracy of self-replication in life. And that's what this paper is about. It was recently accepted for publication in Acta Biotheoretica, which is a mainstream peer-reviewed scientific journal.
So you said that your focus on that paper is about self-replication.
Yes.
And do you actually believe that life can come from non-life without God's intervention, that it can originate by natural means, by self-organization? Or do you believe that God created life and then let life to diversify by itself through evolution? What is your view on that?
Well, I don't have very rigid views. Some of my views have changed a little bit with time. They keep changing, which is actually fairly common in science. But I would say that, first of all, I believe God created everything. The first verses of Genesis make it very clear God created the heavens and the earth, and that includes everything. Now, the details, where the devil lives, of course, I'm not sure. I could go, if somebody figured out that there is a path to the origin of life through purely natural chemical processes, I could be convinced. But I frankly don't think that's going to happen. And the number of problems with an abiogenesis from chemistry to life is just staggering. And it's as James Tour often says, but just the chemistry part of it is extremely difficult to imagine. And I'm focused on what happens. Let's say everything works out great and you have some self-organization and you actually end up with a cell. The cell can't evolve. When people talk about evolution, they talk about natural selection and mutations. But there's another aspect to evolution, which is almost always just skipped over and taken for granted. And that is the question of accurate, very, very accurate self-replication. Nothing in the world self-replicates, nothing. The only thing that self-replicates are living cells, not even chemicals self-replicate.
Yes, I make an analogy which I call the factory maker argument, where I say that cells are chemical factories. And when I bring up that argument, then a very common objection is, well, but factories do not self-replicate. And my answer to that, of course, is that that is actually evidence that I mean, we cannot make factories which self-replicate, because that would be such a complex thing that, I mean, cells, they are working entirely autonomously. They do not require any input or intelligent input or so. So they are working entirely autonomously. So that actually adds a huge amount of argumentation for intelligent design.
Exactly.
And not against it.
Yeah, the story I like to say is, let's say you have a house, and a couple is living in a house. They love the house. They have a couple of kids, and it's in a beautiful neighborhood. It's a great house. Everything they have in it is great. And the kids decide they want a house exactly like that. And so they tell their parents, I want a house right next door, right near you, but exactly identical to everything in this house, including the furniture, the paintings, the books, the clothing, everything the same. Do that. And you could do that. You could hire, let's say the parents say, okay, we'll hire a contractor, we'll get an architect, we'll look for all the same furniture, we'll buy all the same books if we can find them, And you could do that. It would take a long time and a lot of effort. But one thing you cannot do is find a house that replicates itself. And a house, no matter how much you have in that house, no matter how much you have stored in the closet or in the attic, contains nothing compared to what's in a cell. A cell has 1 billion protein molecules. And and that's just the beginning. I mean, all the organelles, all the membranes. And if you look at a cell that replicates, let's say a simple bacterium, and you look at the 2 daughter cells, they're pretty much identical to the original parent cell. And that happens every 20 minutes.
Yeah, so when I investigated the number of proteins in the cell, I was really astonished when I did read that the human cell has 2.3 billion proteins.
That's right.
And I mean, I cannot even imagine such a quantity of proteins in a single cell. I mean, that's just staggering.
That's right. If you hold to evolution, you tend to think that everything that modern life has evolved because it was better. So there was natural selection and it evolved. So the first answer you get is, obviously replication is extremely beneficial. But then my question is, but wait a minute, evolution to work in order to allow these things to develop has to be already include replication. Because the whole point of evolution is, it's not, if a bird gets a better eye and has a better eyesight, that gives him a selective advantage. But if his kids don't inherit that, what good is it? So you have to have accurate inheritance even to start evolution. So how do you get accurate inheritance before those accurate inheritance?
There is a biochemist, his name is Dimitar Kunev, and he also publishes science papers, and we had a few interactions. And his claim is that evolution actually can start prior to DNA replication and when life starts. What would your answer be to that claim and assertion?
I'd like to know how, because if you don't have any replication of a genetic material, you have to have something replicating. Otherwise, I don't know how evolution starts without that. You could have a very good cell, okay? You could have a great membrane with all kinds of enzymes inside that are metabolizing away. You could have an energy generating system, beautiful, great. That cell may live for a long time. It may have a very high probability of survival, but eventually it's going to die, and then you're done. I mean, if it doesn't replicate itself, so how do you get evolution there? I don't get it.
The proposition of theistic evolution, is it that God simply created the chemical elements and left them by their own, and then life also started by chemical evolution? Or is the proposition that God actually created life and then eventually a single life form, and then it diversified, and then evolution kicked in? What is there a consensus about the proposition of theistic evolutions?
Yeah, if you go to the Biologos website, for example, which is the main home of evolutionary creation, which is trying to replace theistic evolution, but it's the same thing. But if you go to the Biologos website and you look for that, what you'll find is that they say, and they're correct, that there's a wide range of views. The key thing to understand here is that abiogenesis is not considered to be part of the evolutionary story. And that's important because we know the mechanism of evolution, but it can't be, at least I believe, it cannot be the same mechanism that would explain the origin of life. And even Darwin, didn't talk about it. Richard Dawkins says very little. He just says it only had to happen once, and then he says a few other things. But in his better books, he says that's not something that is part of evolution. And so you can be an evolutionary creationist, meaning that you believe that evolution is one of the laws of God, basically, like gravity or electromagnetism, but that doesn't mean that you're committed to a natural cause for abiogenesis.
We know that the replication mechanism of prokaryotes and eukaryotes is quite dissimilar. It is different. And there are other points as well, like the membrane of prokaryotes is different than eukaryotes. Sure. And also the ribosome is different. And how do you deal with that?
Well, you're right. The prokaryotes, eukaryotes, and archaea as well have differed considerably in the details of all of those mechanisms. But on the other hand, all living organisms, no matter what kingdom they're in, have the same genetic code with, again, some small differences. That's true. There are a few organisms with a slight difference in the genetic code, but they're off on a branch. So the vast majority of life uses the same genetic code and uses the same general mechanism for replication of DNA, for synthesis of proteins, which is an incredibly complex mechanism. I think that the fact that there are differences is correct, but the commonality of the basic mechanisms of life can be traced all the way back to what we call Luca, the last universal common ancestor.
I mean, I think that the transition from prokaryotes to eukaryotes is one of the biggest evolutionary gaps. And I mean, that would be something that would have to be solved if someone wants to claim that common ancestry is true. And there are actually even science papers which openly admit that the tree of life, starting with eukaryotes and archaea, would be different than bacterial origin. So
right. So let me talk about that. That's an interesting point. The point about, you're right that the prokaryote to eukaryote transition is very unusual in evolutionary terms because it didn't happen slowly and gradually. It happened in one big step. And that was the revolutionary idea that Lynn Margulis proposed, because remember that Darwin originally had said that gradualism was critical to his theory. Yeah. That every change has to, he said that if that could be disproven, his theory would be wrong, which is that every change would have to be itself small so that it could happen and have an advantage, a selective advantage. That's been disproven. One of the things that goes against that, and by the way, that's called the continuity principle. And the reason that's been disproven is because the evidence, which includes engulfing of one cell by another cell, and it also includes, for example, very rapid evolutionary changes that we know occurred based on things like explosion of new species and many other things. So, for example, and just to give one illustration where there's a lot of data, we know that at the beginning of vertebrates, there was a couple of whole genome duplications where the entire genome duplicated itself. Extremely rare event that's happened a few times in the history of life. And that also was a major change in evolutionary, it wasn't any longer gradual or continuous. And that's the basis of the paper that I mentioned. In fact, the title of the paper is about the origin of self-replication and the continuity principle. And the whole point of the paper is that if you're going to have accurate self-replication, you must violate the continuity principle again. So now the question is, what does that mean? When we violate continuity, was Darwin right? Should we throw away evolution? I mean, that's one argument you could make. But Darwin got a lot of other stuff wrong, not surprisingly. I mean, he lived before we knew anything about much. So what you have to do is you have to change the theory to admit mechanisms for these what's called saltational events. In other words, jumps. I think we have to be open to this idea of discontinuity, of gaps. And the endosymbiosis, to me, is an example of that.
One problem is the information, but the other problem is also the assignment of meaning from codons to amino acids, which is also something which I say assignment of meaning is only something which intelligence does. We do not have an example of where unguided events have that capability in principle, actually, to assign meaning, to assign something which means something else. And this is, in my understanding, a conceptual problem. It is, unguided mechanisms simply are not the adequate explanation, and we know only of intelligence which is capable of doing that.
When you ask an atheist, not a scientist atheist, but just a regular atheist, about that question, a new answer has come up. It's only been around a few years, which is that DNA is not really a code.
Oh, yes, I hear that all the time.
You hear that? Okay.
Yeah, all the time.
Rationality Rules, Stephen Woodford, who is a British atheist.
Yeah.
In 2017, he made a video claiming that DNA is not a code. And I made a response video to that, showing his mistake. He gets confused with the terminology. He thinks sequence is code.
Yeah.
But that meme has spread throughout the atheist community. I will see people saying, and these are not scientists, but they'll be saying things. Well, DNA is a metaphor. It's a metaphor for a code. It's not a real code.
Yeah.
That's complete nonsense. It's like a real complete code, and every scientist knows that. It's in every textbook.
Yeah.
So but that's what people do when they're stuck with a problem that they can't answer. They make one up. Look at real science and look at it carefully, and you won't find any arguments against God. There is nothing that contradicts the actions, existence, or presence of God and of Jesus Christ in your life. I mean, those things are not connected to scientific denial.
Well, that is a standard question, which I always ask to atheists. How can we explain our existence without a creator? And there are some which say, well, the gap is shrinking, and the more science advances, the more it will there will be natural explanations. But my reply to that is, I don't know any compelling explanation whatsoever in regards of everything related to origins.
Science is not closing these gaps. More gaps are coming, and some questions will be answered. They always have been, and they will be answered, but not all of them. Having said all of this, it's very important to understand that we can understand the natural world through scientific investigation up to a point, and that point keeps changing. My point is that what we consider hard, definite science and what we consider, supernatural or crazy stuff or whatever, that border keeps changing. That goalpost keeps moving back.
So let's move to evolution side. So you believe that do you believe that the standard explanations in regards of biodiversity, which is the gene-centric view, that still does withstand scrutiny, or what is your view on that? How do we explain biodiversity today? What is the best explanation?
The gene-centric view has come under attack strongly. It started slowly. That's come under scientific attack from many fronts. It's now clear that evolutionary pathways are far more complex than the simple idea of, okay, you have a point mutation in a structural gene. It makes a phenotype that's better for the organism in a particular environment. You get natural selection, boom, evolution. That's sometimes called neo-Darwinian. It's so much more complicated than that. You have neutral evolution, which has no selection, which is very important. You have the Simon Conway Morris's ideas of convergence, where you have the same structures being produced in completely unrelated sets of organisms. You have punctuated equilibrium from Stephen Jay Gould, which says no, it's not a steady, continuous thing, and that's just from the evidence. He was not a theist; he was an atheist. But he looked at the data and he said, yeah, sometimes nothing happens, and then boom, you get this big change. And then you have you have the whole idea of genetic engineering from James Shapiro. You have Kevin Leyland with a niche construction, which is a two-way street between the environment and the genome.
Yes.
I mean, I go on and on. In fact, I've written papers about this on the extended evolutionary synthesis. So, yeah, it's gene centrism is struggling. You have to replicate the entire phenotype of the cell in order to get evolution started. So how do you do that? And the way you do that, you can't do it with RNA, because the RNA world can't do everything. But so how did Luca and everybody else after that do it? They do it by making proteins, making all the enzymes. It's the enzymes that give the phenotype to a cell.
And you need proteins to make DNA replication.
Exactly.
And that brings us to a catch-22 problem.
Exactly. And so you have the DNA which carries the information and gets replicated, but now you have to translate. You have to come up with a translation system, which is what it's called, protein synthesis.
Yeah, the transcription and translation, and you also require already complex proteins to do so.
Yeah. So the issue is, how do you translate nucleotide chemistry to protein chemistry? Because those are two completely different types of chemical molecules. You need an incredibly complex translation system, which includes transfer RNA and ribosomes, and of course these incredible enzymes called aminoacyl tRNA synthetase, which actually translates, and that's where the genetic code originates.
Yes.
But how do you get that? And that has to work well. How do you get that working well without evolution?
Well, I've investigated that, and you have 13 different translation error check and repair mechanisms in the ribosome.
Right.
And in order to have sufficient accuracy in translation.
Right.
And
exactly.
amino-acyl tRNA synthetase, they have internal signaling information transmission where they check if the right amino acid is being added.
That's an incredibly important fact. They have an editing site in the protein, in the enzyme. That's why I started working on this project that resulted in this paper, because what I have done in this paper is I said, okay, forget all the details of the biochemistry. Forget it. We don't know what the details were. Is it theoretically possible, no matter what the mechanism is, for a cell with a very low replication accuracy to survive and evolve? And how high does that replication accuracy have to be? And I did it through a very simple statistical model, which I developed into a computer program, etc. And the results were very surprising and very striking. And that's in the paper. And basically, the conclusion is, this is discontinuity. If you start out with a cell that has less than 65% accuracy in replication, nothing can happen. The cells can't survive. Certainly, they don't evolve. Done. So how does a cell get to 65% accuracy? That's not bad. Okay. That's a pretty accurate attempt, but it's not enough. You have to get above that. So if you have a cell that's 50% accurate in its replication, in its whatever the mechanism is, it's doomed. You can make the argument that some of these perfections came later with evolution, but the question is, if you don't have evolution, how did you do that? And the reality is, I mean, you know what the accuracy is now of DNA replication and translation. Just tell the audience, DNA replication is accurate at 99.99999%.
Yeah, I think it is in 10 billion nucleotides you have an error, something like that.
It's 0.00001% error. I don't, and the same thing is true for the translation system. It's a little less accurate, but it's 99.9999% accurate.
Yeah.
So that if you just take ribosome and RNA and all the ingredients and mush them together, you may get 40 to 50% accuracy. I mean, you need all that other stuff that you mentioned to have anything close to 90, never mind 99.999. And that's true in all modern life. So, there's a big gap to fill. And I think this paper, I hope people read it and understand the point of it. I'm going to be writing another paper that's less technical, that I'll put into a journal, not necessarily for scientists. But I think that this is there's not much answer to this, because this is making no assumptions whatsoever. It's just assuming you could take any system you want, the same effect will occur. And this is just based on statistics.
It was my pleasure to have you at my channel, and I hope that we can keep in touch and advance our conversations. And
yeah, let's do that.
Keep sharing ideas in regards of all these fascinating fields.
Yes, indeed. Okay.
Okay.
Blessings.
Okay. Thank you very much. Thanks, everyone. Bye-bye. See you next time.
Bye.